Polytolypa. Polytolypa is a monotypic genus of fungus containing the single species Polytolypa hystricis. First classified in the Onygenaceae family, as of 2008, it is considered to be in the Ajellomycetaceae, although there is still uncertainty as to its phylogenetic relationships with other similar genera. This species is only known from a single specimen derived in the laboratory from a specimen of dung of the North American porcupine, Erethizon dorsatum, collected in Ontario, Canada. Polytolypa hystricis contains bioactive compounds that have antifungal activity. Taxonomy, phylogeny, and naming. The genus was first described in 1993 by University of Toronto mycologists J. A. Scott and D. W. Malloch, who grew the fungus in moist chamber cultures of porcupine dung collected in Stoneleigh, Ontario, Canada. The generic name Polytolypa is from the Greek word polypolou meaning many, and tolop meaning skein of yarn. The specific epithet hystricis comes from the Greek hystrix or porcupine. The genus has been classified in the Onygenaceae, a fungal family characterized by species capable of digesting human hair in vitro, and with spores that are punctate with minute surface punctures when viewed with scanning electron microscopy. However, as Scott and colleagues demonstrated using traditional laboratory tests to determine keratinolytic activity, P. hystrix is not able to digest hair. There is still uncertainty as to its phylogenetic relationships with other similar genera. Polytolypa is thought to be evolutionarily most closely related to the genera Malebranchia and Spiromastix. The grouping of Polytolypa and Spiromastix represent a sister clade to the Ajellomyces clade based on analysis of partial nuclear LSU sequence data. However, the phylogenetics of Polytolypa are still unclear and await further study. The 10th edition of the Dictionary of the Fungi 2008 considers the genus to be in the Ajellomycetaceae family, although uncertainty with this classification is indicated in the entry. In contrast, the online mycological database MycoBank classifies the genus in the Onygenaceae. Description: the ascues containing reproductive structures, or schemata, are minute, spherical bodies typically 200–400 mm in diameter. They start out white, but gradually become rusty brown in maturity. The schemata, which may be clustered together in groups or scattered about, grow in a shallow layer of hairs, actually fungal mycelia called a tomentum. The schemata have appendages composed of numerous coiled. The ascospores produced by Polytolypa are ellipsoidal, yellow to yellow-orange in color, with dimensions of 2.55 by 3.4 mm. Viewed with a light microscope, their surfaces appear to be smooth, but under scanning electron microscopy, they are revealed to be densely marked with punctures and small, hard, sharp projections. The structures that produce the ascospores are called asci. In Polytolypa they are numerous, spherical, and measure 9–10 by 12–13 mm. Each ascus contains 8 ascospores, which are released when the ascus dissolves away at maturity. The anamorph asexual form of the fungus resembles the genus Chrysosporium. Habitat and distribution. Polytolypa hystricis is known only from the dung of the North American porcupine, Erethizon dorsatum. Porcupine dens accumulate thick layers of nutrient-rich dung, hair, and urine that are degraded by a succession of fungi. These fungi are disseminated by arthropods such as insects or by the porcupine themselves. Bioactive compounds. Chemical analysis has shown that Polytolypa hystricis contains a unique triterpenoid chemical named polytolipin, as well two compounds known previously as metabolites from Scleroderus canker Gremmeniella abietina. Both polytolipin and one of the previously identified compounds have moderate antifungal activity against the species Escobolus furfuraceus, while polytopin alone can inhibit the growth of Candida albicans.